All right, hello, welcome back for your second day of online learning. I hope that day one went well for you guys and that you are getting the hang of this. So today we're going to continue talking about cells, and today we're going to talk about cell types. So we're still on the same standard: conduct an investigation to provide evidence that living things are made of cells, either one cell or many different numbers and types of cells. We do have a few learning objectives today. Let me go back to that one. Let me put this on present so you guys can see it better. Maybe. All right, so our objectives, we're going to learn how to use a compound light microscope. I know that you guys have already had experience with that in the class, but I have some exciting news because I found an online microscope that you're going to be using today. So we'll go over how to use that. You're going to understand that all living things are made up of cells, and you need to compare and contrast a variety of cells in order to see which structures the cells have in common, identify specialized cellular structures and learn how they relate to the cell's function, compare unicellular and multicellular organisms, identify similarities and differences between plant and animal cells, and perform a laboratory experiment to determine if cells and other materials are alive. So cell types. Complex organisms are made up of smaller units called cells, which we've been talking about. Most cells are too small to be seen by the naked eye. Microscopes are used to magnify small objects, so here you will use a compound light microscope to observe the cells of different organisms. And like I said, you already used the microscope in class, so this is kind of just a quick review for you. We looked at all the parts of our microscope. This right up here labeled with an A, that is our eyepiece, and that's the part that you look through when you're trying to look at your sample. This is B, they've got it labeled as B. This is our stage. Remember, the stage is kind of like our platform, and it's going to move up and down as we focus. Then C, we have our coarse focus knob. So you're going to get that in view most of the way with the coarse focus. And then D down here on this particular microscope is our fine focus. So you're going to get it even more detail. Remember, it's kind of like at the eye doctor when they say, which one looks better, A or B? A or B? And then you have to decide which one's more in focus. So you're just kind of focusing in on your specimen really clearly with that fine focus knob. And then we have E. These, remember, are our objective lens, and those are going to give you your different magnification powers. We always start on the lowest objective, and then we move up to our higher objectives. So the last thing they have there is the slide, and we know that we use those in class, and they were glass, and we don't want to break them. But guess what? Online, we can't break them, so we don't have to worry about that. All right, so let's explore a Gizmo. This is going to be a new site that we're going to use a lot through this online platform. And so I'm going to show you at the end of the class how to create your account at explorelearning.com. But for right now, I am just going to show you how to use this. So don't be worrying about how you're going to get there. Just know that you're going to create an account. I'm going to give you a class code, and you are going to be all set to go. So once you do get your account and you go in, you're going to find your assignment, and it's going to be this Gizmo called cell types. Okay, so I'm just going to give you a quick show of how this works. You have this landscape here, and as you see, as I'm moving my mouse around, things start to light up. So anything that lights up can be clicked on, and we can then look at it underneath the microscope. So you can have some fun exploring with this. It's kind of fun. They're going to ask you to find particular things in the activity that you're going to do. So if you don't know what that thing is, you can go ahead and click Show All Samples. Now everything that you can click on is highlighted, and now we can just pick this. So we're going to look at the Elodia leaf. And so once you have it selected, it shows here. Okay, and then you have to switch tabs. So your tabs are up here at the top, and in your assignment, it's going to talk a lot in the directions like go to the landscape tab, go to the microscope tab. That's what they're talking about. These are your tabs. So you're going to click, after you have your item selected, we're going to click on the microscope. And now we have our Elodia leaf underneath the microscope. So these are our adjustments. So instead of actually turning the knob here, we get to adjust our coarse focus just using this dial. And as you can see, when I go that way, it's getting more blurry, right? Because it's lowering the stage. So we want to bring the stage up until it comes into focus. And then we can even get it clearer. Remember that which one is clear, A or B. I think it's nice and clear there, so I would leave that there. Okay, and so now I can take a look at this. You can see clearly that this leaf has cells. All those cells are connected together in there. And this is on the lowest magnification, the 40. So I can now change my objective now that I have it focused in. So now I'm going to get an even better view. And if I want to do a little more fine focusing there, I can try to do that. I think it looked better right where I had it. And then we go to 400. So you can see we get nice and detailed in there at the 400 focus. All right. So these individual chambers that we see here, these are the cells of the leaf. And then it is showing the different parts. You can also click over here to get information. Okay, so this will tell you the cells in an Elodia plant leaf perform photosynthesis, converting carbon dioxide and sunlight from the atmosphere into food and oxygen. Chloroplast, the photosynthetic organelles, give these plants their characteristic green color. And then maybe you're thinking, well, what are the chloroplasts? Where are they? You can also show labels, all right? And let's focus that in just a little clearer so we can actually read our labels. See, now we can actually read what they're talking about here. So these little green, you can see that they are labeled as the chloroplast. And then we have our nucleus, we have our cell membrane and our cell wall and our cytoplasm. All right, so those are just some of the features so that you guys can use this tool. I'll show you a little bit more. We'll do one more with the human skin. It's just like yesterday, we looked at skin and we looked at cheek cells. So if I'm looking for the skin, it's obviously going to be on the person, right? And there it is. Her right arm is going to be our skin sample. So now we have it selected. We go to the microscope. We need to focus it in. That's about as far as I can go. And then we can focus in a little bit clearer here. And again, we can look at it under different magnifications. Continue to focus that. It looks a little blurry. Let's go to 400 and see if we can get it focused in. Oh, let's go the other way. See, if it doesn't work one direction, focus back down the other way. So that looks pretty good. And you can see all the parts are labeled, just like we talked about in our skin cell yesterday. We have the nucleus, the cell membrane, and the cytoplasm. All right, so my labels are on. The other thing I can actually do is turn on my scale. So this is going to measure, so I can kind of get an idea how big these are, okay? So this unit of measurement is actually called a macrometer. And you can't really see it here very well, but it's this funny little U symbol with an M. A macrometer is 1,000 micrometers in a millimeter. So you know when you have our rulers and one of those tiny little spaces is a millimeter? Well, we can fit 1,000 micrometers in a millimeter. So that gives you an idea of how tiny this actually is. And you can kind of use this as a gauge to, they might ask you questions like how long across or how wide is your cell? Well, you could look at this and you could see, well, it's probably, you could space it out with your hands and you could see it's about a little more than 20, right? So that will be a helpful tool for you as they're asking you some questions on your assignment today. You're going to be asked how the size of some different objects, so know how to use that. Or again, just click show scale bars. That's how I got that there. If I want that black line to go away, I just uncheck the box. If I wanted to show me the labels again, I just check the show labels box, okay? And that's pretty much all there is to it. So you're going to have an assignment in your Google Classroom that looks like this: Student Exploration, Cell Types, Part 1. There are some vocabulary words listed up there for you, and I will give you a vocabulary sheet as well. You're going to see that it's just an additional resource that's going to be in your Google Classroom with all of today's information. So please just, you can look at those, just get familiar as you're going through. If they ask you something you don't know, refer to that vocabulary sheet so you can figure it out. So before you even ever go into your Gizmo, please answer these 2 questions. And when you're answering the questions, you can type right in your Google document. You guys know that from before, so everybody has their own copy. So you should be able to type your answers right in here. Then answer these 2 questions because I want to know your thoughts before you actually work with the microscope and look at the different cells. Then you're going to log into your Explore Learning account, which I'm going to show you how to create, so don't worry. So you're going to go into the account and you're going to complete this form as you use the Gizmo. The Gizmo is called Cell Types, and it has been assigned to you. Since I can only see the teacher side, I'm not exactly 100% sure what that's going to look like, but you're just looking for your assignment. So I'm sure there's probably a tab or something in there for assignments, and you're going to click on that. Then you're just going to go through the activities. So here are kind of the directions. The first thing we're going to do is observe cells, and you're going to start by going into your Gizmo. You're going to go on the landscape tab, click on the organelle listed in the table. So here's the table. We're going to start with mouse skin, and then you're going to select the microscope tab, and you're going to observe the samples under the highest magnification. You're going to click the show labels box to label the organelles, and then you're going to list the organelles and approximate size of the cells in each sample. All right, so you're going to list your organelles, and I will split this. I just realized I need another column here. So you'll have a spot to write down what organelles are present in your mouse skin and what size it is. And then you're just going to keep working through. That's the first activity. There's a question. Anywhere you see a question, you need to answer the question, okay? And remember, this is how you're going to get credit for being in science class today, is you need to do this activity using the Gizmo and filling out this form and turning it in Google Classroom. Then you're going to have to observe E. coli. Then in the next part, you're going to compare cells to some sand. We're going to take a look at sand or silt there. And then part B, we're going to look at specialized cells. So we're going to start by collecting some data. You're going to look at the neurons, and you're going to compare some cells here: the human neuron, the human skin, the human muscle, the human blood. So you're just filling out each column as you go through those. Then we're going to observe human skin. You're going to find that you can click on each label. Oh yeah, I think I forgot to show you that part. So when you're in here and you have show labels on, if you click on the label, it tells you exactly what the function of that part of the cell is. So pretty cool. If you click on one like over here, cytoplasm, and I can't read it, it's outside this circle, right? So just remember that you can move your slide just like in real life. Remember, if you want it to go right, you have to move it left. If you want it to go left, you have to move it right. Same thing here, but these are your stage left and right, and this would be your stage up and down. So it's going to move your cell this direction. Okay, so go ahead and look up the functions of these parts as you go through this. And then the last part, you're going to observe the human neurons. My computer's lagging. Okay, we're going to compare red and white blood cells. So in the slides, they show you which ones are red blood cells, which ones are white blood cells. And then this one's pretty fun. You're going to compare humans with animal samples. So you're going to start out comparing the human and the worm, the neurons. You're going to do the human and the fly muscle, and you're going to do the human and frog blood. And you're going to be, just give me what you see in general. Do you see many differences? And what organelle do frog red blood cells, RBC stands for red blood cells, have that human red blood cells do not? And that is it. When you finish this activity and work through all those pieces, fill in your answers, and then you're going to submit this, of course, through Google Classroom. All right, so now I'm going to show you how I want you to set up that count in case you haven't done it. I did send this out to you guys earlier in email so that you guys could set this up or through Class Dojo. So here are the simple steps. You're going to go to the Explore Learning website. You're going to click on Enroll. Actually, the actual button says Sign in /enroll. So enroll. Then you go enroll in a class. Oh, sorry. Woo. And then you're going to have a code. So your code is going to be on your sheet that was emailed to you, or it's right here. So if you don't know where that sheet is that got emailed to you, or you didn't get it, or whatever the case is, you can find your code. So here's my class, period 0, period 1. Here's Mr. Crawford's, period 2, and period 6. I would like you, when you create your password, to please make it the same as your Google Classroom login information. That way you do not have to remember it. You'll already know it, right? So this is just my example, john.smith@durellacademynv.org. Oops, I forgot the student. You guys are student.durellacademy. And then just use your student number. My little thing is covering that up right now. There we go. So your student number, I don't know, whatever your number is, make that your password, okay? All right, so let's review your next tasks. What you need to do. You need to create your Explore Learning account and enroll in your class if you have not done that yet. You need to open up the Gizmo, and the assignment is cell types, and it's been assigned to you, so open that up. Then you need to open the Google Doc, the one I was showing you, the March 24th Student Exploration Cell Types Part 1. You need to complete the Google Doc and submit it by midnight tonight in order to be marked present in Science for Today. Remember, you do not get marked present for being in class unless you turn in your completed assignment. If you just turn it in and nothing's there, you didn't do the work, you do not get credit. You need to turn in a completed assignment before midnight in order for me to mark you present in class today, March 24th. All right, guys, thank you for coming and watching the video. Good luck on your task. If you need help, please contact me through Google Classroom or through my email, and I will be in open office hours from 9 to 9:40 and 10 to 10:40. I can't remember exactly. One of those is 6th grade, one is 7th grade. You have the office hours sheet. Okay, so come see me, and I will see you later.